reflex inhibition through the overriding influence of spinal interneurons. So your brain might be going, okay, like go crazy, let's activate this muscle. But if you get excessive muscle tension forming, it'll again override through this reflex, which goes straight onto the alpha motor neurons and inhibits that production. So it's a productive sensory mechanism to protect the muscle and the surrounding connective tissue from injury. And this is what I thought we were going to get at with, not the next slide, but the slide after, but it turns out it's not. We're going to do it anyway. Okay, last sensory information reflexes, the muscle chemoreceptors. So these muscle chemoreceptors send sensory information on the metabolic state of the muscle to the cardiovascular and respiratory control sensors via type 2s and 3s nerve fibers, so the afferent nerve fibers. So your muscle, basically, the point of these last 3 slides is that your muscle has a whole bunch of receptors in it that are not only telling the brain what's going on in this area, depending on like the stretch, the tension, the metabolites. So these are linked, they're going to go back to the brain and let it know what's going on, but these are also going to have reflexes to help control your muscle activity. Your muscle chemoreceptors are sensitive to changes in hydrogen ions, carbon dioxide, oxygen, and play an important role in the regulation of cardiovascular and respiratory responses to exercise. So this is something that's obviously not conscious. You're not really going to know what your metabolic milieu is during a exercise, but this information is going to go up and through these central integration sites in the brainstem cause efferent responses in like your heart rate, respiratory function, all the stuff we're going to talk about in our 6 lectures together. Yeah?
And that's what I mean, like for example, with the PCO2, like the metabolites accumulate, you're going to hyperventilate.
Mm-hmm.
That's okay.
Exactly, yeah. Exactly. Okay. So I found this lecture a little bit dry, and I also thought that when we were talking about the inhibition of muscles, I thought of something that would be really cool to demonstrate it, but then I did a little bit of research. So we're going to do an experiment. We're going to do Constan's phenomenon. So this was first described by a German neurologist in 1915, and I need 2 volunteers. It's fun. I promise you won't look like an idiot. You'll feel real cool. Ah, yeah! Okay, I need each of you in a doorway. So face us, stand in the doorway, and look out at the room. Has anyone, do we know what we're going to do here? Oh, I'm so excited. We did this like when I was in public school. Okay, so what you're going to do, I also need someone to time 30 seconds. Does anyone have their phone out? Got a timer on it? Okay. Yasmin's going to time. So what you guys are going to do is push out on the door frame as hard as you possibly can without stopping for 30 seconds. And then at the end of 30 seconds, you're going to step out, and we're going to see what happens. Don't spoil it for them. Okay. Ready? As hard as you can. 3, 2, 1, go. So while they push, I really thought that this was going to be something about like the Golgi tendons or something like that. But spoiler, people didn't really know, and they've been doing research on this, which was published in like 2014. So they recently, what are we at? Keep pushing hard. They were looking at actually what mediates the magic that we're about to see.
4, 3, 2, 1.
Okay, step out. Oh, you did it!
Well, how
Did you stop it? Did you? What happened? Nothing?
Nothing about my arms, still kind of funny.
They went, yeah. They did a beautiful one. Okay. So basically, the idea. Nice job, guys. That was beautiful. Okay, so the idea is that your arms kind of fly up in the air afterwards. So there was a study. I was trying to figure out what mechanism it is to just get someone to link it back to like the Golgi tendons or something like that. And I found this paper, which is from 2014, if you want to read it, our aspiring motor neuron person. Does anyone want to guess how it works? I'll tell you that I had to read this like 4 times. I only read the abstract, but I thought I would give you guys the opportunity to guess if you want to. All right. So the paper concludes that there is, in the brain, a persistent generator of involuntary motor commands. So basically, when the voluntary, if they, and this might have been what happened, Harlan. This might have been what happened with Harlan, was that, I don't know, but and actually this happened with Sid too, that her arms were going up and then she brought them down, because they said that if they had their subjects, and they were doing a whole bunch of stuff like TMS and stuff on them, if they told the people, so as their arms are coming up, to try to not let the arms go up, they could override the involuntary motor command to bring the arms up. So it could come down, but then as soon as they said, okay, stop trying to force your arms down, then the arms would just go back up again. And they had this involuntary arm lift resumed. So their results, this is a direct quote from their abstract, suggest that voluntary motor inhibition may be a specific neural function distinct from the absence of positive voluntary motor commands. So adding another layer and sort of underscoring the idea that all of this motor neural control of motor functions is still being figured out. There's some stuff that we know for sure, like the size principle. I haven't just been telling you guys random stuff today, but there's a lot in this area. This is like an area of ongoing research. I have friends who research this, and yeah, if anyone's interested. Yeah, so it's a good thing we did all those demonstrations because we are early. Does anyone have any questions? You guys just want to go, right? Okay, so I'll see you guys in a week or so, I guess.